Okay, we're going to start with what you need to know for muscle types, and then what you need to know for muscle anatomy. So for muscle types, I want you to know the following. First, voluntary, involuntary. Obviously, when I talk about muscle types, I mean skeletal, cardiac, smooth, those three. Voluntary, involuntary. Cell structure. Branched or non-branched or spindle-shaped. Striations. Yes or no. Basically, smooth muscle does not have striations. Number of nuclei. Okay, we're good? Possible stimulus. Skeletal muscle only. Nerve signal, the smooth and cardiac nerve signal, chemical, mechanical, or hormonal stimuli. Does that make sense so far? Okay, good. Next, what to know about muscle structure. Basically, we're talking about levels of organization. So, at the level of the whole muscle, it is surrounded by epimysium. So that's connective tissue, okay? Muscle as a whole, like your biceps or triceps or whatever muscle we're talking about, consists of multiple fascicles. Okay? I'm basically going from the biggest level to smaller and smaller and smaller. Does that make sense? So fascicle surrounded by perimysium, and then muscle fiber, which is essentially the muscle cell, is surrounded by endomysium. Structures to know within the fiber. Sarcolemma, which is the membrane, cellular membrane. Sarcoplasm, which is the cytoplasm of a muscle. Sarcoplasmic reticulum, which is basically endoplasmic reticulum. So far we're good? Just terminologically. T-tubules, extensions of the sarcolemma. We're going to talk about them, don't worry. Myoglobin, oxygen storage. It's a protein, it stores oxygen. Glycosomes. Responsible for glycogen storage. So far we're good? And now I'm going to project some structure of next thing over here. Myofibril. So I'm not going to write down what it is. I'd rather talk about it. So this is, imagine that this is the long rod. Okay? like this. Okay? Long rod. This rod can shorten, basically, it can contract. When several myofibrils contract, the muscle fiber contracts. When muscle fiber contracts, the muscle contracts. Does that make sense? So I deliberately made my representation of myofibril from several markers, because myofibril consists of individual contractile units called sarcomeres. Okay? And this is what we're going to talk about now. So we're going to look at now when I say contractile unit, it means the whole sarcomere contracts. When you have these multiple sarcomeres lined up in a myofibril, when they all contract, myofibril contracts as well, and the muscle contracts, and so on and so forth. Does that make sense? Basically, we can look at the mechanism of the contraction using one sarcomere only. So let's do it. At the center of the sarcomere is the protein structure called the M line. Okay? It's very convenient to color code this. Okay? Sarcomere is bound by so-called Z discs. So within the Z discs is a single sarcomere. Does that make sense so far? Now M line has thick filaments attached to it. So these are thick filaments. They are composed of the protein myosin. Now you can imagine that if there are thick filaments, there's going to be thin filaments. So thin filaments that consist of a protein called actin are attached to Z discs. Well, to make it more precise, they're not attached directly. Okay, so there are little elastic filaments here. We're not going to pay too much attention to elastic filaments on the functional level. So protein that forms them is called titin. Okay, elastic filaments attach thin filaments to Z discs. Okay, now how does the couple more structures here? So you can see that there's a region right here in the center where thin and thick filaments kind of overlap. Can you see that? So that region is called A band. Now, these regions here, where there is no overlap and there is only thin filament, each one of those is 1/2 of I band. So you see, while A band lies completely within one sarcomere, I band, half of it is in one sarcomere, another half is going to be here in another. They are organized like cars of the train. Are you following me? So A band is darker because of the overlap. I band is lighter. This is why we see striations. And striations, remember striations, dark light stripes on the muscle cell? Basically, striations tell you this muscle cell contains sarcomeres, so it contracts by the means of sarcomeres. You follow me? So like cardiac muscle and skeletal muscle, they are both striated, which means there are sarcomeres in both. Smooth muscle is not striated. It does not have sarcomeres. Okay? So how does the contraction happen? The myosin protein has kind of two parts. It has a heavy chain and it has a light chain. So heavy chain is the structural component. It forms the filament. The light chain forms so-called myosin head. Okay? Yeah, not the greatest picture in the world, but okay, I can live with it. So these are myosin heads. You with me so far? So what can myosin head do? This structure, the head of the myosin, it can bind to actin, which forms thin filament. Well, on my image it does, right? And it also can break down ATP. What is released when you break down ATP? Say it out loud. Energy, yes. And we need energy to move, right? So, since it can break down ATP, you're going to move. Now, what happens during contraction? At the level of the sarcomere, we're going to dive even deeper into that. At the level of the sarcomere, look what happens. These myosin heads, they propel actin thin filaments towards the M line. Does that make sense? If not, I have an example. This ruler is the thin filament. My arm is the myosin head. During contraction, this happens. Okay? Literally, to the M line. So sarcomere, it's called sliding filament mechanism. Filaments do not change the length. Look at me. My arm doesn't change length, right? The ruler does not change length, but they slide against each other. Okay, we good? Make sense? Okay.